In this video, we're going to investigate how DNA is replicated in order for it to be divided into new and growing cells. By the end of this video, you should understand why, where, and how DNA replication occurs. First, I need to tell you that before cells divide, in the process of mitosis or meiosis, both of which are cell division, DNA has to be copied or replicated. So that replication process has to happen before any sort of cell division can occur. As a reminder, we know that in eukaryotic cells, DNA is housed in the nucleus. And so this DNA replication in eukaryotic cells occurs in the nucleus. And when we talk about DNA replication, we call it semi-conservative. And you guys should know that prefix semi. You've seen it in semicircle, semi-truck. It means partial or half, right? And then conservation or conservative means saved. So DNA replication is literally half saved. And what that means is that one strand is copied or conserved, and the new strand is then added to that other side. So we call this semi-conserved or semi-conservative. And we're going to talk and see a little bit more what this looks like. So here's a really great illustration of what that semi-conservative DNA replication looks like. Here in the blue, we can see original DNA strands, and so that's the parent strand, the original strand. Then what happens is we get these newly synthesized strands. Those are the red ones you see here. And the reason that DNA replication is called semi-conserved is that you can see there's a blue original template and the newly synthesized red joining together. So let's look at a more in-depth version of this. So here's a really great illustration of DNA replication over time. We can see in the beginning our DNA parent strand is blue. We go through our F1 or first generation, and you can see that this is our original strand and this is our new one. You can also see that over here. Then when we move into F2, you can see that DNA is also replicated again, and this red strand is here and the new red strand is joining. So we can see over time that these DNA strands, this original blue DNA strand, never goes away. It just moves further and further down those generations. So how does DNA replication happen? Well, first, DNA is held together by hydrogen bonds, and these hydrogen bonds need to be broken in order for new strands to come in. Those bonds are broken by this enzyme. Remember, an enzyme is a protein that speeds up chemical reactions. So these hydrogen bonds are broken down by an enzyme called DNA helicase. Pretty much, they're molecular scissors, and they cut that DNA molecule right in half to unzip the DNA molecule. Then the DNA is in separate halves, and those halves are complementary, meaning one side would have an A and the other side would have a T because those two sides correspond. So here we can see this happening. Right here, that's our hydrogen bond between our two sides of our DNA, and over time that DNA helicase unzips that DNA and creates two separate but complementary sides. Then what happens is nucleotides match up with complementary bases. So here we have a C, we know a G must come in, an A, and a T, and T and an A. So these nucleotides, one by one, come into the DNA and form the complementary base pairs. Once those nucleotides are placed, they're the light gray ones here, then DNA is zipped back together. So it combines that DNA back together and reforms hydrogen bonds. And this is done by an enzyme called DNA polymerase. DNA polymerase also proofreads for copying errors to make sure that A and T are actually matched together and C and G are matched together. So here is a summary of what we've talked about. Here's our DNA. DNA helicase breaks these hydrogen bonds and unzips our DNA molecule. Then we have two complementary strands. Now that our strands are separate, our nucleotides can come in and match to those old strands, and then DNA polymerase comes back together and joins those sides with hydrogen bonds. That DNA polymerase also proofreads this process. Then we're left with two identical DNA molecules The light gray is our new strand. The dark gray is the original strand. So here is just another picture of DNA replication to really hit home what happens. Our old strand is broken in half. These new bases of nucleotides are added, and then we have an old strand and a new strand, which, remember, that's why DNA replication is called semi-conservative. So, like I said, DNA polymerase normally checks for copying errors, but sometimes it misses those copying errors. And these copying errors are called mutations. And mutations are a change in the sequence of DNA nucleotide bases. So for some reason, something does not get copied properly, and it creates a mutation or an error. And we're going to talk a lot more about mutations and errors in DNA replication as we move on through this class. So now, at the end of this video, you should understand DNA replication. You should be familiar with the enzymes that help that process happen and the steps that occur.